Hello everyone, welcome to my channel. So this is the final part of sexual reproduction in flowering plants, and today we will be talking about post-fertilization events. We have already discussed how is the formation of microspore, how is the formation of megaspore, and double fertilization we have already spoken about. So, in the description of this video, I will be giving a reference to all the previous videos that is there of this section. Okay, so we are talking about post fertilization events. So, when I talk about the post fertilization, once the fertilization occurs, it should be clear to us that the ovary forms the fruit, the ovules forms the seeds, and I have the integuments which forms the seed coat. So now going ahead, what happens? We already talked about the double fertilization. We had a triple fusion that occurs between the polar nuclei and the sperm cells or the male gametophyte, one of the male gametophyte. So there from the primary endosperm cell, there is a mitotic division of the nucleus that occurs and we get ample number of free nucleus. So these free nucleus is later on, cell wall formation is there, it is celled up, and hence we get the cellular endosperm. So this free nucleus is being used up by various cells, and it is being taken up for the embryo, and we get cotyledon. So we have a free nuclear type, and then we have got a cellular type of endosperm development. Among the cellular, we have a non-albuminous endosperm, which completely utilizes the endosperm while the maturation of seeds are taking place. Example, pea, groundnut, all the things. You will get thick cotyledons, and albuminins are those which uses only a portion of endosperm while the maturation of seeds like wheat, maize, castor. So you will have something juicy inside. All right. So if you have ever taken maize, you will find that there is some syrup inside, or wheat, rice, anything like this. Now, if just coming into a very good example of making you know what is an endosperm is coconut, the tender coconut that we usually have. It is known for a very good amount of electrolytes inside it. It is also known for the nutritious value. So the free nucleus, I told you, this endosperms, they are a good source of nutrients. It permits, or it allows the maturation of seeds. Hence, it is a completely nutritious matter that I have in the endosperm. So the liquid part of the tender coconut, the water that you have, is nothing but the free nucleus that you have. That is the liquid endosperm. And the cellular endosperm is nothing but the white part or the kernel that you have, that is known as the cellular endosperm. So this is the endosperm that you have. So these endosperms, they will be maturing into seeds. So these seeds may be monocot if they are having a single cotyledon, or maybe a dicot if they have two cotyledons. So if like maize have a single cotyledon and hence called a monocot, and if I have the pea, they will have two cotyledons. Groundnuts, they will have two cotyledons, and hence I call it as a dicot. So what is the difference between monocot and dicot? So there's one cotyledon in monocot, there are two cotyledons in dicot. Monocots are basically have fibrous roots, they have parallel veinations. If you see a maize plant, if you would have seen a maize plant, you would have seen bhutta, plants of bhutta. You will have, they are basically fibrous and they have the parallel veination which is there. And they have a scattered stem vascular bundles. So the flower parts in trees. So there is a group of three petals that is there and that is in the flower. In dicot, there are two cotyledons. They have a tap roots which is there, and there is a net leaf veination. That is the network reticulate veination is there. And then we have vascular bundles. They are in the form of a ring, and flowers in the form of 4 or 5 that occurs. Coming to the structure of monocotyledon seeds, we have the monocotyledon seeds, if you can see. So there is an aileron layer which is there. Inside that, there's an endosperm. This endosperm, I told you, this is albuminous seed. Hence, only a part of endosperm is utilized for the maturation of seeds, and the rest remains. So there's an endosperm which is there. The basically in the maize, the yellow part that you see, that is nothing but the endosperm, and the white part that you see, the center, that is nothing but the embryo. So therein I have the scutellum, which is the cotyledon which is there, and then I have the plumule that will grow up to shoot, and covering the plumule is the coleoptile, and there is a radicle which will form the roots along with the root cap, and the covering of the root cap is coleorhiza. So I have coleoptile is towards the top, tile, you can understand it's towards the top, so coleoptile and coleorhiza. Now in a monocot embryo, in the grass family, basically the cotyledon is called the scutellum, which is situated towards one side, which is a lateral of the embryonal axis. At the lower end, the embryonal axis has a radicle and root cap enclosed in an undifferentiated sheath called coleorhiza, and the portion of embryonal axis above the level of attachment of the scutellum is epicotyle. Epicotyle has a shoot apex and a few leaf primordia enclosed in hollow foliar structure, which is called as coleoptile. Now coming to a dicot embryo. In a dicot embryo, I have two cotyledons, and lower part I have the radicle, which is covered with a root cap, and above the cotyledons I have hypocotyle, which has a plumule in it. So there are two cotyledons which is there. You can see like in peanut that is there. And here we had the embryo very much on the top, which forms the radicle and the plumule. Now, coming to a seed, the seed, it's a fertilized and mature ovule which develops into seeds. It consists of cotyledons. It may be monocot, it may be dicot. So it has an embryonal axis which is there, and the seed coat double layer which is formed by integuments. So there are two seed coats, one which you can remove out like in peanut or in chickpea. There is a seed, there is a coat which you just soak it in water. In you can go ahead and peel it off. So that is the testa, and then one very thin inner coat is there which is almost attached. That is known as a tegument. So there's a testa and tegument which is there. The seed coats are there. And then I have a micropyle. Micropyle, I told you, where the entry of the pollen tube will take place. There's a small opening on the seed coat. It facilitates the entry of water and oxygen into seeds for germination. Okay, so this is the micropyle which is there. Below that, there is a small scar on the seed coat, which is the hylium. Okay, There's a hilum which is there, which is a small scar, which you have a small hole like matter that you will see. So that is the hilum. And then you have the seed, which may be albuminous or non-albuminous seeds. Then after that, we have a perisperm, which is the remnant of the nucellus that is present. That is in black pepper. In this, you will have some amount of nucellus, which is already present. It gets grounded and all those. And then there is the seeds. They will germinate if they get enough amount of proper nutrients. They get sunlight, they get water. So before they germinate, those seeds we can germinate later on. I say the seeds are dormant or the seeds are sleeping. They would wake up at a later point of time. The wall of the ovary, they develop into a pericarp. So there are different kinds of fruits that we have. We have a true fruit. True fruit means it originates from ripened ovary. From the ovary fetus forming, I call it as a true fruit. But if it is originating from the thalamus, so I call it as the false fruit. It is not from the ovary, but from the thalamus. If in a flower, you remember various parts are there. So there was a thalamus at the end. So that if it originates from the thalamus, then I say it's a false fruit. We another have a parthenocarpic fruit, which actually goes ahead and it germinates without any fertilization. So the condition in which fruits are developed without the formation of seeds, this is mainly due to absence of fertilization, pollination, and embryo development. So this is where we have the parthenocarpy, which is there. Mostly bananas without seeds are parthenocarpy. The next part, we have another two ways of development. One is apomixis and polyembryo. So apomixis is nothing but is asexual mean of seed production where embryo develop from cells of maternal genotype. From the mother cells, it will go ahead and there will be a development of seeds. And they are not a product of any genetic recombination or syngamy. So syngamy is not occurring. So it's like what it is like, it's a asexual reproduction that is occurring, but it is trying to mimic the sexual reproduction. That is the seed is formed without fertilization. So that there is a formation of seeds, but there is no kind of fertilization that would occur. Okay, like if you say in the sexual reproduction, what is happening? Uh, you know, Meiosis will occur. Then after that, there in the ovary there will be the formation is there, and then the sperm will go ahead. There's a formation of endosperm, and they go ahead. But here, what happens? Here, the megaspore mother cell, they will go ahead, and there's no reproduction will occur. So there is no reduction and no recombination will occur. So there's a 2n which is there. Okay. So reduction is not taking one mitotic division, and then there is a formation of egg cell, and there is no fertilization that will occur. Hence, the I'm getting a maternal clone, just a clone of the mother. So there's no recombination which occurs in apomixis. So formation of apomictic seeds, which is that they are diploid cells formed without meiosis. They develop into embryo without fertilization. They have cells of nucellus, which is 2n, surrounding embryo sac, protrude into embryo sac, develops into embryo. Example, we have citrus and mango, all those things. Next, we have polyembryo. When I have in the integuments, where I have a numerous embryo. So occurrence of more than one embryo in a seed, we have citrus or groundnuts, which is there. So often this is associated with apomixis. So we have a polyembryo, which is there. We have the integuments, and I have the integument embryo throughout. So that is there I get. So now to differentiate between apomixis and polyembryo, apomixis is asexual reproduction that occurs without fertilization and not involving meiosis. Polyembryo, the formation of more than one embryo from a single fertilized ovum or in a single seed. Apomixis is asexual reproduction. Polyembryo is a sexual reproduction. In apomixis, fertilization does not occur, and in polyembryo, it undergoes fertilization. In apomixis, it does not involve in production of seeds, whereas in polyembryo, it involves in the production of seeds. In apomixis, the offspring's plants are genetically identical to the parents, whereas in polyembryo, the offsprings are not genetically identical to the parent. So that's it, and thank you.